thanks for the introduction and welcome everyone. I'm very happy to once again have a chance to present our work at KIDS, and I'm very excited that we can have this meeting as planned, just in a virtual form. So today during my talk, I will try to answer a question if we can solve the unsolvable with a SNP panel that combines ancestry and phenotype markers. So first I would like to refer to a paper that was published in the end of last year that summarizes the current state of phenotyping in Europe. So for those who are interested in the topic, this paper walks you through all of the available commercial and non-commercial solutions for phenotype and ancestry predictions. And what is really important, it also explains the legal situation in Europe because it varies between the countries. From those where phenotyping is forbidden or not specifically regulated, to those that already implemented new legislations and now allow to use phenotyping in practice. So one of them is Germany, where such a change happened in the end of 2019. And now a forensic specialist can be asked to analyze casework for phenotype and biological age of an unknown individual who contributed to the trace. And we have a special situation in a federal state of Bavaria, where my institute is located, where such a law exists already since mid-2018 in an extended form, because it also allows for ancestry predictions, but the typing can be done only in special cases, for example, to avoid serious danger. So in our institute, since the end of 2018, we are looking closer how introducing MPS would change our routine work. And with the platform that we have, observing the changing legal situation and minding the available commercial solutions, we decided to go for all-in-one phenotyping kit. So together with Thermo Fisher and the White Glove team, we designed an ION-AMP-seq SNP panel. The final version includes 196 targets that cover 202 autosomal hotspots that are associated with ancestry and phenotype, and the mean length of the amplicons is around 80 base pair. Additionally, we also added some Y-chromosomal markers in the final number of 120. They are covered by 130 targets, and the main length is around 200 base pair. And they cover 20 major haplogroups from the ISOC-Y phylogeny, and additionally, we have 100 subhaplogroups for possibly better phylogenetic resolution. So to sum up, those 41 markers we see here, they built a panel known as HighRiskPlex S that allows us for eye, hair, and skin color predictions. The rest of the autosomal SNPs are known as Precision ID Ancestry Panel for the ancestry predictions. And as already mentioned, we also have the Y SNPs to understand better the male lineage. So to test our panel, we performed sensitivity study for which we selected a known male sample that we diluted from 1 nanogram to 7 picogram and amplified it in triplicate. The libraries were done manually according to guidelines for custom ION-AMP-seq panel. The rest of the workflow was done by ION-Chef, and the samples were sequenced on ION S5 with 5 digits. And this is the workflow we use generally for all of the rest of the samples. So all of the results were reviewed in Converge, but the ancestry and the phenotype markers were analyzed separately because the software cannot perform phenotype predictions. So we used it to export the genotypes, and then we translated them in Excel to get the input that is required by HighRiskPlex as web tool. So here are the results of the phenotype predictions based on the consensus genotypes consisting of between 40 to 24 SNPs at 7 picogram, showing a decreased accuracy starting with 62 picogram. In case of eye and skin color, all of the inputs had the same prediction, namely blue eyes and pale to intermediate skin, and this was in concordance with the actual phenotype of the individual. And for the hair color, all except one samples had light blonde predicted, One sample at 31 picogram had the prediction for brown color, and this was caused by the fact that we had in one SNP wrong alleles code that passed the thresholds for coverage and heterozygotic balance. The actual color of the hair was blonde, dark blonde, but we know that the individual was light blonde as a child. So for the final interference of ancestry, we first calculated the admixture, and they were predicted in Converge. So the software uses a data set that includes genotype frequencies and population data for 145 SNPs out of 165. And the method that Converge is using is called bootstrapping method. So a random set of sequence SNPs is analyzed multiple times, and an average results of all of those replications is presented in the end. The admixture is presented as a percentage of each population with a corresponding likelihood, and we have 7 populations included in Converge, which is Europe, Southwest Asia, South Asia, East Asia, Africa, America, and Oceania. So here you can see the ancestry predictions. The number of SNPs used was between 144 and 107 at 103 at 7 picogram. So the predictions were correct for all of the inputs, suggesting European origin. We also used the same SNP set, and we ran the estimations in Snipper. They were also all predicted as European. And in fact, our sample came from Germany. To enhance the ancestry predictions, we also calculated population likelihoods using FROG. And we decided for the lately published standalone version that allows to analyze multiple genomes at once. Our SNP set was tested on 96 populations. And the highest likelihood that we got calculated was Irish, mixed Europeans, and Danes. As mentioned, our sample was German, but Germans are not included in a database. So for the last part, the Y-chromosome, for the haplogroup prediction, it was the same down to 31 picogram, and this was the most derived SNP within the panel. And below that, we only had the major R haplogroup prediction. We also used NavGen and the YSTR data to estimate the haplogroup, and we confirmed the predicted lineage. So the second stage of the validation was testing the panel on known reference samples. And they were collected based on an agreement with an ethics commission from our university. And we asked volunteers to fill out a questionnaire when they self-declared their ancestry down to the third generation and described their phenotype. And we took reference pictures for comparison. So for this study, we collected and used 124 samples from Europe. 84 of them were from Germany. We also had 11 non-European samples and 5 samples that were known to be admixed. So here the outcome of the phenotype predictions for all 140 samples done using the guidelines for high-risk S. For eye color, 3 results were inconclusive, and the rest were predicted as blue or brown, and 88 of those predictions were correct. For hair color and skin color, we had predictions for all possible colors, and accordingly, 78 and 89 of them were correct. So those values overall correspond with the values obtained during the validation of high-risk S. So here you can see a graph that presents that mixture predicted by Converge for European, non-European, and the non-admixed samples. So if we have a look at this in detail, here we can see the European samples. They were all assigned correctly to Europe, some of them showing admixture mostly with Southwest Asia. On the right, you can see the results from Snipper. All of the samples were predicted as 100% European. Just as a comment, the reference set that we used in Snipper didn't include the samples from the Middle East. We also calculated population likelihoods that didn't bring too much information, as Europeans are pretty complex population. But what we did observe that for almost all of the samples that had that mixture, European mixture, at least 90%, we had major European populations available in the data set with the highest likelihoods, like Irish, Hungarians, mixed Europeans, or Danes. And the samples that had some Southwest Asia admixture very often also had Turkish population or ethnic groups like Ashkenazi Jews having the highest values. And when it comes to the Y haplogroups, all of them were atypical European haplogroups and also were the particular lineages here. Now the non-European samples. So we had altogether 7 Asian samples. We had the East Asian that both by SNPer and Converge were predicted correctly. Also the population likelihoods here were all East Asian. And for 3 of the samples, the highest one even corresponded with the actual ancestry of the individual. Then when we look at the Southwest Asia samples, they were admixed, showing a high percentage of Europe and Southwest Asia. And by SNPer, they were predicted as only European or only South Asian. And the populations that were calculated here as the highest likelihood were all Middle East. And even if the actual population was in the database, it never got calculated with the highest likelihood. Then for the Africa, we had an example of East, Central, and North African sample. So only one sample was assigned to Africa, the one from Central, both by Converge and SNPer. When we look at the East African sample, it was admixed with Southwest Asia, and the sample from North Africa, which actually came from Egypt, was assigned as Southwest Asia only. And when it comes to the population likelihoods, they corresponded with the intracontinental origin of the individuals. And the last sample was from America. It had a declared Jewish ancestry, and it was predicted as 100% European. So now we are moving to the admixed samples. So here are the results from the admixture prediction from Converge, and above is the expected admixture based on the questionnaires, so down to the 3rd generation, so the grandparents. So we had example, for example, so this was always an admixture of a European individual and non-European individual. We had a situation where we had admixture with 2 North African individuals, and the difference between those admixtures and the admixtures that we got before for the declared Europeans was that we observed a higher admixture of Africa that we didn't see before next to the Southwest Asia. And in one case, we had a male for which we also predicted the wild haplogroup, which was J1A, which is rather infrequent in Europe, and this corresponded with the North African ancestry of the individual. We also had an interesting example where we expected admixture of South America and Europe, and what we got predicted was Europe and Africa. So it is known that there are many African lineages in South America. In this case, the South American lineage was female. So we will be looking closer at the sample also with the mitochondrial DNA to understand it better. And now we are moving to the last part, the most interesting one, which was studying Casework samples. So this was a blind study where the Casework samples were unknown because the person who was interpreting the result didn't know their origin. We had 16 samples that were collected during the autopsies performed in our institute, 14 blood samples and 2 bone samples. Then we were also able to test our panel on actual traces collected at the scene and submitted by the Bavarian police for phenotyping. So we had different input for the samples and different degradation index. We studied all of them in duplicate. So this table here, it's a sum up, a complex sum up of all of the predictions that we were able to perform and all of the results that we got. In the gray box, you can see the bone samples. They were the most degraded ones. And you see that for one of them, we didn't have the phenotype prediction, and also we are lacking the Y haplogroup estimation. For all of the rest of the samples, we calculated the admixtures, we calculated the population likelihoods, and predicted the Y chromosome haplogroup. And additionally, what is on the slides, but unfortunately I don't have time to go into details, is the mitochondrial haplogroup that we also predicted based on the control region that we are allowed legally to sequence for those kind of cases. And this slide is a sum up of all of those results coming together. So the predictions that we did according to the final phenotype and the ancestry. So first, the concordance with the reference data when it comes to the phenotype. In the green box, you can see all of the phenotypes that were actually correct because we had the reference data coming from the autopsy. For the rest of the samples, the data was lacking because the bodies were too decayed or skeletonized. And when it comes to the ancestry, the information provided by the police was a place of birth of those individuals. And based on this, all of the predictions were correct, except of one. For the individual that we predicted to be European, both based on autosomal markers and haploid markers, as a place of birth had stated Brazil. And now the really last part of the study, which was testing those actual traces. In the end of last year, we got our first case for phenotyping from the Bavarian police. But back then, our panel was still being validated. So the samples were also sent to Innsbruck, which was running Visage panel on it. So we had two traces from the scene. These are the results from the first trace, and these are the results from the second trace. Based on the SDR, we expected to have the same contributor to both of them, and we got exactly the same predictions for both traces. And what is the most important for us, also those results for the ancestry and for the phenotype were concordant with the results from Visage. So can we solve the unsolvable? I think that this is a very complex question that still needs some time to be answered, but each study in the field of phenotyping brings us closer. And what we learned from our research is that when we combine all of those predictions together, if they can really shed light on unknown individual, very important and informative are the haploid markers, also the ones that we added into our panels of the Y-SNPs. We definitely think that we need more samples to study in the future to understand admixtures even better. And we already plan to use the gathered and sequenced genotypes to provide the missing frequency data for German population. Thank you very much. In the end, I would like to acknowledge everyone who helped with this research because, of course, lots of people helped with it. Thanks to GMI, especially Walter Passon, who discussed with us the concordancy on the results that we got. Thanks for Temo Fischer, who are really, really helpful with designing and going through all of the details with the custom panel. And definitely my team, without whom I would not get those results today. So thank you very much.